Hi everyone, today we're going to do an experiment to explore how different molecules move through a selective permeable membrane. We'll use a dialysis tubing to represent a cell membrane and see what happens to starch and glucose over time. This is called a qualitative test. It is a test that tells if something is present or there, but it does not tell how much of the substance there is. For this experiment, we'll need the following: glucose iodine, which is a special liquid used to test for starch; glucose solution, a simple sugar dissolved in water; starch solution, Benedict solution, which is a blue liquid used to test for glucose, sugar; and a dialysis tubing, which will act as a cell membrane. Take a moment to observe. Beaker 1 contains water. Later on in the video, you will notice that the color changes to a brown. This is because we will be adding iodine to it. In the beaker, you will notice a bag that is the dialysis tubing. The tubing contains a solution mix of starch and glucose. Pause the video and take a minute to record your initial observations of the color of the liquid in the beaker with the dialysis tubing. Make sure you also record your observations on the substance inside of the dialysis tubing. Now let's look at beaker number 2. We have a beaker with water in it and 3 test tubes. Test tube number 1 has glucose or sugar. Test tube number 2 has starch. Test tube 3 has water. Pause the video and take a minute to record your initial observations of the color of the solutions inside of the beaker and the test tubes. Now let's focus on the beaker with the test tubes. You will notice that the color inside of the liquid inside of the test tubes has changed. This is because we added 2 milliliters of Benedict solution in each of the test tubes. Pause the video and record your observations on the change of color. We then place the beaker with the test tubes on heat and wait for it to boil for about 5 minutes. Make observations about what you see or any changes that occur during this time. So you're supposed to have one already done. Let's focus on the beaker and the dialysis tubing. This is where we will be testing for iodine. You may have noticed that as we heat up the test tubes and wait for a change, we prepare a beaker with the dialysis tubing in it. Inside the dialysis bag is a mixture of starch and glucose. The beaker contains water and iodine, so it turns brown. Record any observations in the change of color on your observation sheet. We will let the dialysis tubing sit in the beaker with the iodine liquid mixture for 30 minutes or until we notice a change. Remember that the dialysis tubing is acting as a cell membrane. While you wait for results, make a prediction. Do you think there will be a change in color? If so, which molecules do you think will move and where will they go? While we continue to wait for a change in the dialysis tubing, let's focus back to beaker number 2. This is where we're testing for glucose with Benedict. Pay attention to the test tubes with the solution. Look at the color. Has it changed? You can notice that the beaker solution changed from a bright blue to a pinkish red. This shows that glucose was present in the solution. Don't forget to record your observations on your data sheet. While we continue waiting for results on the dialysis tubing, we will prepare beaker number 3. Let's check if iodine is present with starch. Pause the video and record your observations on the initial colors of the solution in each test tube. So iodine
in that. So you're going to put.
Now, I will be adding iodine to each test tube. Pay attention to any changes in color. Make sure you record your observations. If the solution turns an amber or brown color, it is negative for starch, meaning there is no starch present. Make a prediction. Do you think it will be positive or negative for starch? If the solution turns dark blue, black, it means it's positive for starch. If needed, pause the video and record your observations. As you can see, the test tube with starch did in fact change colors. It turned a blue-black, meaning that it tested positive for starch. The other two test tubes did not turn a black color. Now, looking back at the dialysis tubing, do you notice any changes? Write down your observations. Notice that the color did change in the dialysis tubing. It now looks a purple color. This means that iodine is present inside the tubing. How did it get there? Well, as the dialysis tube soaked in the iodine solution, molecules moved through the tiny openings in the tubing, similar to how substances move across a cell membrane. We can also notice that starch stayed inside the tubing because the solution in the beaker did not change. It is still an amber or brown color. But how does this happen? This experiment models how selectively permeable membranes work, just like cell membranes in your body. The dialysis tubing acting like a cell membrane. It is selectively permeable, which means it allows some molecules to pass through but blocks others, kind of like a filter or a strainer. Glucose or sugar and iodine molecules are small, so they can easily pass through the tiny holes in the tubing. However, the starch molecules are too large to fit through the holes, so they stay inside of the tubing. Think about this. If our cell membranes let every molecule pass through freely, what might happen? Those cells could potentially be destroyed. That's why membranes control what goes in and out. Now that we've completed the experiment, take a moment to record your observations, evidence, or any questions that you may have on your student recording sheet. Thank you for watching and great job thinking like scientists.